So thus we come to the end of the discussion of the basic structure. So we will go somewhat interior to the structure of interior into the structure of retina.
Discuss something about the retinal cells in this particular video. So retinal cells are slightly more complicated than you think it is, since the retina is having around 10 layers. That particular simple single small structure called as retina is in turn having 10 big layers inside it, and each layers are formed by multiple cells. So retinal cells are slightly difficult to learn, but in our level it is much more easier. We are supposed to learn only 3 important cells. Okay, so just learn something about retina over here. Retina is the neuronal layer. It is the modification of a nervous system. You have a nervous system inside your body. It is made up of neurons, the central nervous system, peripheral nervous system, autonomic, somatic, whatever it is made up of neurons. Even the brain and spinal cord are also made up of neurons, including retina. Retina is a part of nervous system. It is a modification of nervous system. So whatever cells are there in retina, they are also neurons. But what is the difference in those neurons? Those neurons are slightly modified enough to transmit visual impulses. They are slightly modified neurons which are modified enough to transmit visual impulses, right? Now you can see the word I have written over here is histological. Histology, what is the meaning of histology? The word meaning of histo means that we are talking about tissue. The study of tissues in the body is called as histology. You take a small section of retina somehow, I don't know how you can take it, but after you are read, the eye can be taken out, you can take the retina, you can take a small sample of that, you can make a slide with this, and then it is seen under the microscope. And once you see that under microscope, it will look very complicated, but you can make out 10 layers. Okay, the number of layers you can just remember from this. If they won't be asking any of these kind of things, but if you want to remember this, it is good and well and good. Remember, it has 10 histological layers. 10 layers. Okay, 10 layers are there in retina. The cells of the retina, this is much important, very important, that you have 3 important cells in retina. You have multiple cells in retina, but out of that we will be remembering 3. That is how you need to remember this. Just because you have to learn just 3 cells doesn't mean that the retina is very simple. It is complicated, but you have the syllabus tells you to learn only 3 cells. And those cells are photoreceptor cells, bipolar cells and ganglion cells. Three important cells over there. Photoreceptor cells, bipolar cells and ganglion cells. Now, which is the outermost of this? Photoreceptor cells are outermost. The light falls on the photoreceptor cells. The light falls on retina. Where exactly in the retina? In the photoreceptor cells. Now, from the photoreceptor cells, what happens is the impulse is transmitted from the photoreceptor cells into the bipolar cells, and from the bipolar cells it is transmitted into the ganglion cells, right? From the photoreceptor cells the impulses are transmitted into the bipolar cells, and from the bipolar cells it is transmitted into the ganglion cells. So which is outermost? Photoreceptor cells. Which is innermost? Ganglion cells. Bipolar cells, it is very easy to remember, it is in between. The word between starts with B, and bipolar cells also starts with B. So you can learn it in that way. Bipolar cells are in between. So outermost is photoreceptor, then you have bipolar cells and you have ganglion cells. Never ever forget this. They can ask questions like, which is the first layer that light encounters? Photoreceptor cells. What is the pathway of visual transmission within the retina? Visual impulse transmission within the retina. You will say photoreceptor cells, bipolar cells and ganglion cells. Right? So these kind of questions are multiple times asked in your practice test, and I expect these kind of questions to be asked in the NEET exam as well. Very probable questions these are. Now, the ganglion cells are the innermost cells. Okay, so these ganglion cells are the innermost cells. I said that they are neurons. If they are neurons, they will be having axons, they will be having cell body, etc., etc. These axons will be collectively joining. They will be collectively joining. All the ganglion cell axons will be collectively joining to form the optic nerve. See, take a look at this picture. You have this particular cornea over here. Light passes through the cornea into the aqueous chamber. You have aqueous humor over here. Then you have a iris. This is the iris. It has 2 types of muscle: radial muscle, circular muscle. We have learned that. And through this, it goes into the lens. Okay. So through the lens, it goes into the retina. And inside the retina, this is the vitreous chamber as such, and this is the big vitreous chamber. Through this particular vitreous humor which is present inside this, it will be finally landing on the retina. Where exactly in the retina? The photoreceptor cells. And then through the ganglion cells, it will be forming the optic nerve. So it passes from the photoreceptor cells into the bipolar cells, and finally the ganglion cells it will reach, and these ganglion cells, axons, they will be forming the optic nerve, and this is the optic nerve. You can see it is going through this, and the central red line is the artery. Okay, so this is an artery which is supplying the eye, so it is coming from the brain as such. Okay, it is coming from the brain. Interesting point to remember is, just for your curiosity, let me tell you, from the heart it goes into the brain, and from the brain it comes out through the eye, through the optic nerve. It is going to come out. This is called as the central retinal artery. Okay, so this is called as the central retinal artery. Just remember, it is coming into the center of retina, almost the center of retina, through the middle of the optic nerve. So the mid, I mean, if you have an optic nerve is a fibrous structure. In the mid part of this particular optic nerve, it is a neuronal structure. You can see an artery coming here, and that is called as the central retinal artery, one of the very important blood supply for the eye. It is covering the majority of the eye structures through that, and that goes into the choroid. Choroid is the vascular layer, isn't it? So that is how we can understand this. Let's quickly go into this. See, this is how the whole structure is. If you can look at the right side of this particular image, this is how they have drawn it very beautifully. So these are the photoreceptor cells. Okay, see, photoreceptor cells, these green things are the photoreceptor cells. They will be passing the impulse into the bipolar cells, isn't it? So this is a bipolar cell, and from the bipolar cell it goes into the ganglion cells, and this ganglion cell axon, these are the axons, they will collectively join to form the optic nerve, and that goes into the brain. Where exactly in the brain? Finally it will be reaching the occipital cortex, the back side of the brain. So you have photoreceptor cell, bipolar cell, then the ganglion cells. What are the other cells? Other cells, you can see bipolar cells are there. This is the horizontal cell. Then you have an amacrine cell, Muller cell, etc., etc. Nothing to worry about that. Don't care about this at this particular moment. Become a doctor and learn about all these kind of things. Become a medical student and then you can learn about all these kind of things. You have all the time in the world to learn all these kind of things once you get into MBBS. Okay, so this is how the whole thing will look like in the histology. Histology means you take a section, make a slide with that, and stain this with a stain called as hematoxylin eosin stain, and you see it under the microscope. You will appreciate that. If you just talk about the photoreceptor cells, see, this is the cell body of the photoreceptor cells. They are neurons. Mind it, they are neurons. These are the nuclei in the cell body, and this nuclei are stained purple here. Okay, so these purple things are the nuclei, and the other structures are the other processes which are stained pink. Okay, these are the pinkish structures over here. Now, the two types of photoreceptor cells are rods and cones. You have been knowing this since your lower classes, from your lower classes. So this particular photoreceptor cell looks like a rod, and that is why it is called as a rod. And this is having a conical projection over here. That is why it is called as the cone.
And the photoreceptor cells are of two types. One is called as the rods, RODS, and the second one is the cones. So rods and cones are the two types of photoreceptor cells present in the retina, and they are responsible for two types of vision. So if you are viewing anything on the daytime, so it is the cones which are responsible for you to see anything in the daytime when there is light. So that is why we call it as photopic vision. So photo means light, and rods are responsible for the purpose of scotopic vision, that means at night time. So this is seen during daytime, this will help you to see during the night time. Cones are responsible for photopic vision, rods are responsible for scotopic vision. So that is how the whole types of cells are discussed over here, isn't it? Now, for the purpose of vision of an object that you see, you require certain pigments in these kind of cells which are photoreceptor cells. They contain certain pigments which will help in the purpose of vision, and those pigments I have mentioned over here: rhodopsin, iodopsin and melanopsin. These are the three important pigments present in the cells. What cells? Photoreceptor cells. Where? In the retina. Rhodopsin is present in the rods, right? You can very easily remember it from the name itself. It starts with R, the rods also starts with R. So you can link it to that particular aspect and you can learn that rhodopsin is present in rods. Iodopsin is present in the cones, right? And this is called as, rhodopsin is called as the visual purple. See the color with which I am writing it, it is called as the visual purple, and iodopsin is called as the visual violet. and there are three types of iodopsin, right? Three types are there. One is for blue color, one will be responsible for the purpose of viewing the green color, and the other will be responsible for viewing red color. What about all other colors? Only blue, green and red are viewed by the iodopsin, but all the other colors can be made by mixing these kind of colors. So it will go for such a kind of interaction for you to see all the colors in the world. And when all the cones are stimulated, when all these three things are stimulated together, you will be having a mixing of all these three colors, and you will see the white color as such. And melanopsin is very different from rhodopsin and iodopsin. So rhodopsin is visual purple, iodopsin is visual violet, rhodopsin is present in rods, iodopsin is present in cones. But in certain other photoreceptor cells in the retina, you don't have the presence of rhodopsin and iodopsin. Instead, you have a third different pigment that is called as melanopsin. See, melanopsin is very easy to remember, because if you can ever relate it to the word melatonin. See, melatonin is the word to which you can link it, and you will get the function of melanopsin. See, those kind of cells will detect whether it is daytime or night time by the light that is coming into the eye, and when this is stimulated, it will tell you whether it is the time to sleep or it is the time to get wake up. So it is actually telling us something about the circadian rhythm. It is maintaining the circadian rhythm in the human body. So this will maintain the circadian rhythm. Sleep wake cycle is maintained by it, and also the pupillary light reflex. If you just shine a light into the pupil, what happens is that if bright light is if you shine bright light into someone else's eye, you can see that the pupil will get constricted. And if you shine some dim light, now if you stop shining the light into the person's eye, you will see that the pupil will dilate. So that is also mediated with the help of melanopsin. So two functions of melanopsin: one is that it maintains the sleep wake cycle, second it maintains this pupillary size also. So those are the two important processes, right? Now all these pigments can be divided into two parts. The structure of these pigments, if you see, it is actually divided into two parts. One part is called as opsin, and the other part is called as retinal. Now as this particular retinal, the word itself is suggesting that it is a compound which is formed of vitamin A. So this is a compound which is formed out of vitamin A. It is a vitamin A derivative. Retinol, retinal, etc. are the other names of vitamin A. So retinal is the aldehyde form of vitamin A itself. Al for aldehyde, you have learned it in chemistry, isn't it? So it is the form of vitamin A. It is the congener, or you can say it is a form of vitamin A, and the other thing is called as opsin. Now among all these three structures, rhodopsin, iodopsin and melanopsin, the structure of opsin will be different. That is how you get three different kind of pigments. And iodopsin has again three types: one for blue, one for green, one for red. Now that is how you get it as rhodopsin, and all other visual pigments are made up of this particular opsin and retinal, where retinal is type of vitamin A. It is a structure which is formed by vitamin A itself. So let's proceed to further details. Continuing with the discussion of retina, see, on the retinal surface, on the posterior most part, see, in the back side of retina itself, if you see, I have just made a circle here representing that retina from the most back side aspect. If you see that back aspect of retina, you can see that this is the pole, that is the center of retina. See, this is the centralmost point of retina, that is called as the pole of the eyeball, or pole of the retina itself. Now actually I showed you in the previous image that an optic nerve is actually moving away from it, isn't it? It is coming out of the eyeball and it is going to the brain. Where exactly in the brain? On the posterior most surface, on the occipital lobe of the brain. There only vision is actually understood, vision is processed. So on the pole of the retina, that means on the most central aspect of retina, that is not the area where the optic nerve is leaving. So it is discussed in the textbook that from the pole you need to move medially. Medially means you have to move towards the nose. See, this is my medial aspect, isn't it? This is the median aspect, that mid part of the body. So from the pole will be somewhere over here, From there I have to move somewhat medially. See, I have just moved some area medially and I have to move somewhere above. So I will just come here and then I will go up to see the optic nerve which is going away from this. So optic nerve is moving away from the eyeball, optic nerve opening, that is called as the optic disc itself. This optic disc is actually at a location which is medial to the pole and above the pole. So medial and above is the location of optic nerve. Now this area is much important over there. Now this area where the optic nerve is leaving the eyeball, there are no photoreceptors. That means photoreceptors are rods and cones, isn't it? So if there are no photoreceptors on the surface where the optic nerve is leaving the eyeball, you will not be able to see anything if the light is actually falling on this particular aspect where the optic nerve is leaving. And this area is called by the name of your blind spot. So this is the blind spot. You are blind over here. So if at all a light ray comes and falls over here, since this is the area where there are no rods, no cones, you will not be able to see anything. That is why it is called as blind spot. Now, what is there on the pole of the eyeball? What is there in the central most part of the retina? If you just magnify what is there in the pole, I will see that there is an area, disc-like area, which is called by the name of macula lutea, isn't it? So this is called as macula lutea, and in the center of this disc-like area, I have a area that is called as fovea centralis. If you just look at the name itself, fovea, so there is a depression, and there is a depression in the center of macula lutea. That is why it is called as fovea centralis. Now, what is the interesting fact about fovea centralis? There are only cones. So there are no rods over here. Cones are present, but rods are absent. Now, it is in this area that the visual acuity is maximum. So it is the only area in the eyeball with the most maximum visual acuity, that is called as fovea centralis. When you are reading your textbook, each word will be falling on your fovea centralis, and that is how you can read it very clearly. So this is the area for most clear vision, where cones are present, only cones are present, no rods are present, and it is thickly adhered. These cones are actually thickly adhered to each other. It is the densely packed cones which are present over there which is responsible for the highest visual acuity. And remember one more point here, that since this is a depression over here, that it is a depression in the surface of retina, this is the thinnest point of the human eyeball. So thinnest part of the human eyeball, or thinnest part of retina, is the fovea centralis. Right. So with this, I think I have completed everything with respect to the structure of human eyeball. So let us go into the mechanism of vision after this.